Alrighty, hello everyone. We're going to be looking at asexual reproduction today. So, with our asexual reproduction, so the things that I'd like you to get out of today's lesson, of course, is from the key science understanding from our subject outline. So, this includes the fact that cells arise from pre existing cells and that cell division leads to an increase in the cell number. Cell division occurs in somatic cells, and this is different from those that are produced by gametes and germline cells. Continuity of life relies on the replication of genetic material and transfer to the next generation, and this can be through the process of either binary fission, mitosis, meiosis, and fertilization. And then, of course, the products of binary fission and mitotic division have the same number and type of chromosomes as the parent. So these all are in the Essentials Workbook, Chapter 2.6. So, of course, let's get started on looking at the continuity of life. So, the continuity of life, as we said from our topic understanding, is that it requires cell division. So, cell division is pretty much a result in an increase of cell number. The result of cell division is the production of two daughter cells that are identical to the parent cell. And of course, prior to cell division, the DNA is replicated, providing two identical sets of DNA. So that part's really important. And we've got one set, of course, for each of the new cells that are produced. But for our prokaryotic cells, of course, they divide by something called binary fission. So if we have a look at our binary fission, um, binary fission is a form of asexual reproduction. So this is a production that is found or used by prokaryotic cells. So prokaryotic cells, they are simpler and they're less specialized than our eukaryotic cells. So, of course, we know that they've got no nucleus, they've only got one circular chromosome consisting of double stranded DNA, as well as smaller molecules of DNA known as plasmids, which you might have heard a lot about when you were doing your first topic, DNA and proteins. So, like I said before, it is the process that is carried out by our prokaryotic organisms to be able to produce itself. So binary fission, the same as other types of our mitotic division, it will produce two identical offspring. Um, so here we've got a bit of a diagram. So we've got ourselves our chromosome in the middle here. So that's another way of talking about our genetic material. We've got the cell wall around the outside, the plasma membrane on the inner, and then we've got our cytoplasm. So we can see here that there is no nucleus. We can see that it's replicated itself. So I've now actually got two strands of my chromosomes. The chromosome here is attached to the cell membrane, and we can see this division that's slowly occurring here that then results in our two daughter cells that have the identical pieces of genetic material. But to kind of formalize this process a little bit better, our main steps in binary fission is that DNA replication occurs prior to the process occurring. There's no point in starting the process and realizing that we actually don't have the genetic material required for the other cell. So this occurs first, and replication results in those two identical DNA molecules. Um, Once this occurs, we've got ourselves a condensation of DNA to form two identical circular chromosomes. So condensation just means that it's starting to, I guess, coil up and get a little bit thicker. Um, each of these chromosomes attach to the inside of the cell membrane, which we did see before. And we noticed that our prokaryotic cell will actually begin to elongate or get a bit longer, which helps to separate the chromosomes and moves them to opposite ends of our original parent cell. So they're getting longer in the process and kind of separating out due to that elongation. And then a new cell wall and membrane form, and the cell divides into two identical daughter cells, each with their own bit of DNA. So this should be in the figure 263. I don't believe it's page 225. It might be. It could also be maybe 230. So there are advantages of our binary fission. So the first one being that this can occur at a really rapid rate. So I don't know if you know, in terms of when we talk about our bacteria, we know that they populate or are able to grow really quickly. This makes them a really good source for us to be able to use for things such as, looking at the effects of, could be anything, DNA replication. Maybe we're looking at effects of adding a certain type of um, antibiotic and then trying to see, the effects over time because they reproduce so quickly. E. coli is a bacteria that's usually used often in labs. I use that a lot in my lab as well. So reproduction, like I said, occurs rapidly. Okay, it has very exponential growth. So this is figure 264 on page 230 of your workbook. Here we go. This is like as before, useful for using bacteria for a specific process. So of course, copying of DNA, connecting to genetic engineering concepts. You might have been looking at using those plasmids as a way to be able to manipulate DNA and insert those plasmids into DNA strands. I'm sure you would have looked at that in your first topic. Of course, there are some disadvantages, though, of having the exact same copy of yourself over and over again with absolutely no different genes that have occurred. So, because the offspring are identical, it means that they are going to be affected the same as the parent cell. Okay, so thinking about whether or not they are all in a climate where we've taken some antibiotics, luckily for us, they're going to die, but then it means when that bacteria is reproducing, it means that they're also going to die because they have the same genetic material as each other, so they're going to react the same way. And then, of course, this have to be in terms of antibiotics, can be in terms of just the environment itself. So think about global warming, think about climate change, all the things that are changing our environment and making it a bit more stressful for things such as our prokaryotics as well to survive. So that's a bit of a disadvantage there. So because they are all genetically identical, they're going to respond to change in the same way. Cell division by mitosis. So this is essentially how eukaryotic cells are able to arise from our pre-existing cells. Okay, so we know for prokaryotic cells, we just went through binary fission, that's how they are able to keep reproducing, and that's how they pre-exist from existing cells. Whereas with our eukaryotic cells, we use both mitotic division, which is what we're going to go through today, and also meiotic division, which we're going to go through in another video. So mitosis is necessary for the formation of a fully developed adult after a zygote has been fertilized. A zygote, again, we'll go into more detail about what that is, but that's when a sperm and an egg pretty much come together to form a little tiny zygote that then becomes a fetus. And then, of course, for ourselves as eukaryotic organisms, we find that mitosis is really good to be able to replace and repair damaged cells and tissues. So think about when you get a really bad sunburn and you become really red and you start peeling. That's actually that peeling off of your skin is actually your cells rapidly shedding and trying to repair themselves because sunburn damages your cells. Okay, so your cells are literally trying to regenerate themselves because you've damaged them and they're trying to repair that tissue. Eukaryotic cells, of course, like I said, this is their version, I guess, of dividing and making themselves into more versions. Same as binary fission, we produce two identical daughter cells, and we can kind of see that at the end here we've got two daughter cells formed. So this process, of course, is called mitotic division, and then this consists of both mitosis, which is this section here, followed by cytokinesis. So that is our process that we're going to be going through. So mitosis involves the phases of prophase. We've got prometaphase, less commonly really used, but it's there. Metaphase, anaphase, and telophase. Now, back when I was in school, I was always told to remember this process as IPMAT. So in brackets, P M A T. So the I, the interphase, of course, here, it's on the outside because it's technically not a part of mitosis. So IP, PMAT, and then we've got this bit here inside the brackets that is actually what's involved in our mitotic division, as well as cytokinesis at the end. So, of course, I've done it right over this. Mitosis is followed by cytokinesis at the end over here, which is the division of the rest of the cell. So this bit here, mitosis, this is division of our genetic material. Cytokinesis, this is the division of everything else in our cytoplasm, including all of our organelles. Okay. So interphase, this very first phase, remember I put the I outside of the brackets of the PMAT. This is the phase of the cell cycle when the cell's not dividing, it's just living, it's doing everything else that a cell does at this time. So just a bit of terminology here, just to help us out before we go into mitosis. So prior to mitosis, DNA in our nucleus, of course, is uncoiled strands called chromatin. So these are our uncoiled strands, and that's just to really allow for easier transcription and DNA replication. Okay. Um, After DNA replication occurs, remember we need to have two sets of our genetic material so that it can actually divide into two cells. Okay, we can't just have one and divide that into half. That's not going to work. So in interphase, this is where we begin to have that DNA replication to produce two sets of chromatin. So um, two sets being that it forms these little two things here. And we call these two identical bits of DNA, we call them sister chromatids. This whole thing is a chromosome. The two separately are sister chromatids. Okay. And they're attached at the center by what's called a centromere. And it's these two sister chromatids that will separate and be in their own new cell at the end of our mitotic division. So, in summary, interphase this is the non-dividing phase. It's where growth, synthesis, everything else occurs when it is not dividing. So it actually spends most of its time in interphase. Okay, that's probably the biggest part of the cell cycle. It's interphase. It's everything else. Okay, mitotic division is, takes up a portion of the cell cycle. That's just really where it's starting to renew itself. So prophase, this is where we find that the nuclear membrane and nucleolus disappear. And then our chromosome and spindle apparatus appear. So the spindle apparatus is really important. It is part of the cell's cytoskeleton, and it's used to try and move around those sister chromatids or the chromosomes. In metaphase, our chromosomes line up along the equator of the cell. So think about our Earth. We've got an equator. The equator goes halfway through the Earth. So just think the equator of the cell is just, I guess, the middle point or halfway through the cell. Anaphase is when our chromatids move to either end of the cells. Remember, it's a chromosome is when I've got both of my sister chromatids together. So now if I'm just talking about a chromatid, that's one half of that chromosome has now been moved to either side. In telophase, we've got our nucleoli and nuclear membrane reappear, because remember in prophase they disappeared. And then our chromosomes and spindle disappear. And then, of course, we've got our cytokinesis that follows this last phase. So the images that you'll need to follow are on pages 230 to 231. I did update that. Of the workbook to describe each of the stages of mitosis. So please make sure that you've got labeled diagrams that are in your notebook for you to refer back to. However, we'll be going through our different stages in more detail. So interphase, like we said before, and I'm going to keep saying it, this is where your cell's just doing everything else. Please don't think of interphase as a phase where it's not doing anything. It's just not, going through mitotic division. It's just not dividing. However, this is the bit where DNA replication does occur. So this is where they start creating an identical copy of itself ready for that mitotic division stage. In this case here, our chromosomes are not visible, so we can't actually see them here. So at the moment, we've actually only got, like we said, chromatin, that uncoiled pieces of DNA. We've still got our nuclear membrane, we've still got everything else. Centrioles, so these are the little things here. This is where our spindle fibers are generated from. And they are what control the contraction of the spindle fibers during anaphase, so when they're pulling apart those sister chromatid. So for our non-dividing cell, chromatin is condensing into chromosomes. This is where they're starting to wind up around those histones and becoming chromosomes. And the centromeres have duplicated in preparation for mitosis. Okay, so even though this is not an official stage of mitotic division, stuff does occur here to prepare it for that stage. So in early prophase, we can see here, we can actually see those chromosomes. They've actually now become visible because they've condensed by coiling up around histones. So we can actually see them now through this. I'm going to assume this is an electron microscope. Um, or it could be a light microscope. I don't know. Depends. Um, condensation. Condensation is necessary so that the DNA can easily move around the cell. Okay, I think if they're uncoiled pieces, it's kind of like trying to pick up a tiny piece of hair and trying to move it around. I mean, it is possible, but it's a bit more difficult. So at least if it's more coiled up and more visible, it's easier for those spindle fibers to grab onto and move around. In this case here, we've actually no longer got a nucleus membrane. This is broken down now. So now these chromosomes are actually just chilling in the cytoplasm where all the other organelles are. And then we can see here our centrioles start to move towards the poles as they get ready for mitotic division. Okay, so these centrioles are moving to the top pole here and the bottom pole here. In prometaphase, so just another way that we can call it late prophase, this is where we've got our spindle fibers. These have begun to form. And just a side note, spindle fibers are just made of microtubules. So in the process of these forming, we've got our kineticals of chromosomes attached to the kinetical of the spindle fibers. So here are our kineticals, or really, I guess the word that we're more so going to use is the centromere. Okay, so we've got the spindle fibers, they're attaching to the centromeres of our chromosomes. In metaphase, this is where officially all of our chromosomes line up vertically along the equator. So remember the middle of our cell, same as Earth, it's the middle. This here we can see more formally is our chromosomes also lining up on the equator of this cell. And remembering that our spindle fibers, okay, we can see here, we've got that the centrioles and the spindle fibers. So the spindle fiber is attached to either side of our centromeres. Okay, it's really important that we've got two attached on either side. Okay, otherwise it would mean that if, say, this one wasn't here, this whole chromosome would be pulled to one side, and we'd no longer have the ability to have one of these sister chromatids be on either side, which means that one of our cells will have more DNA than it should, which isn't good. So we need to make sure that they attach to either side to make sure that they pull apart. So an anaphase, this is where they actually contract. So remember, the spindle fibers contract and they pull those sister chromatids to separate ends of our cell here. So we can kind of see here it's going to get pulled either side here. And in this more graphic version, we've got the sister chromatids getting pulled apart to either end. Okay, so we can now say that there are genetically identical sets of DNA on either end of our cell at the moment, which is always nice. So our very final stage here, so it's kind of just another way of thinking it's the reverse of prophase. So rather than our nuclear envelope and nucleoli disappearing, they're now beginning to reappear and form around, well, the nuclear envelope anyway is forming around our two um, separate bits of DNA, and our nucleolus or nucleoli, plural, are appearing. So now that the genetic material has been replicated, our chromosomes will start to uncondense. So they will begin to no longer be raveled around those histones and will no longer be able to have them be visible anymore. Don't forget, in prophase, we said that the spindle apparatus began to develop. Well, now it's actually being broken down. So in a plant cell, we can see we've got our prophase. Remember, this is where our chromosomes become visible, nuclear membrane breaks down, spindle fibers begin to form. In metaphase, this is where our chromosomes are lining up on the equator of our cell, so the middle of our cell vertically. In anaphase, their sister chromatids are getting pulled apart through the attachment of their centromeres that are then attached to our spindle fibers, and they're getting pulled apart to either end. And then in telophase, again, our nuclear membrane gets developed or formed, as well as the nucleolus, though I can't really see it in this image here. Our chromosomes then uncoil, so they are no longer visible, right? I think from here to here, they're no longer visible anymore. And what we can see here, this is a cell plate. We're going to go through this in the next few slides, but this is this process of the cytokinesis where we're dividing the cytoplasm. So cytokinesis in animal cells, we've got this cleavage furrow. It's kind of this pinching action, like pinches to be able to halve or cut off our cytoplasm from the one big cell to two smaller ones. When this is occurring, it's not just the cytoplasm that's being divided. Of course, it's also all the organelles. They're getting divided out into our two new cells as well. In plant cells, it's a little bit different. Instead, we've got a cell plate that forms. So we've got all these different vesicles that start to form this cell plate. But of course, remember, plant cells, they also have a cell membrane, so they also have to produce a cell membrane as well. So cytokinesis in plants, like I said, we've got our vesicles containing membrane components fusing to form our cell plate, and then later on they'll go and form that cell membrane. And then in our animal cells, we've got the cleavage furrow. So that contracting or contractile ring that's kind of essentially pinching that cell in half. And then we can see here getting pulled apart more until eventually they will just cut clean off from each other. And that's all there is to be had about asexual reproduction. Next video, we're going to look at meiosis.